SDS page, sodium disulfide polyacromate gel electrophoresis, is a technique used to separate proteins based on their molecular weight. It involves denaturing proteins with SDS to eliminate structural difference, ensuring separation is based only on size. The protein migrates through a polyacromate gel when an electric field is applied. Smaller proteins travel faster while larger ones move slower, creating a size-based separation pattern. SDS is an anionic detergent that binds to protein, giving them a uniform negative charge. This eliminates differences due to protein shape or charge, allowing size-based separations. Polyacromate gel acts as a molecular sieve, slowing down larger proteins while smaller proteins move more easily. The electrical field moves proteins from the negative cathode to the positive anode ends. Protein size can be compared to a molecular weight marker, which is protein ladder. The instruments used are power supply that provide an electrical current to move proteins, gel casting apparatus used to prepare the polyacromate gel, electrophoresis chamber holds the gels and buffer for running the samples, pipette and tips that used to load protein samples into the gel wells, protein ladder as marker, a reference for estimating protein size. SDS page buffer solutions include the running buffer, stacking gel buffer, and resolving gel buffer, and staining solutions such as co-massive beryllium blue or silver stain to visualize separated protein. Protein samples extracted from various sources such as cell lysate, purified proteins for analysis, serum or plasma used for clinical application, recombinant proteins for research and biotechnology. SDS page is an essential technique in biological and medical research with several important applications. First is protein analysis that helps identify proteins' purity and molecular weight. Secondly, for biotechnology and medicine, used in disease diagnosis, vaccine development, and protein research. Thirdly, quality controls ensures proper protein expressions in recombinant protein productions. Lastly, research applications that often used in combination with Western blotting to detect specific protein.
Okay, so next we're going to talk about gel casting process. The first step is tape both ends of the gel tray using laboratory tape. Make sure the edge of the tray is sealed securely. Place comb into the comb guide on the gel tray. Make sure to put the gel tray on the flat surface. Pour molten agarose that has been cooled at 55 degrees Celsius into the gel tray. Next step, pour the agarose into the gel tray until it comes to within 2 to 4 mm of the top of the teeth of the comb. Next, remove the comb by pulling it straight up and gently when the gel is cool and turn opaque. To use the gels, remove the tape and remove the gel from the wells before you use it.
I will now explain the sample preparation process for SDS page. First, add SDS page sample buffer to the protein sample. This buffer contains sodium dodecyl sulfate SDS, which denatures proteins and imparts a uniform negative charge, allowing them to migrate based on size during electrophoresis. The next step is add a reducing agent such as dithiotridol to break disulfide bonds and further denature the proteins. The third step is heat treatment. Incubate the sample at 95 to 100 degrees Celsius for 5 minutes to ensure complete denaturation. The fourth step is centrifugation. After heating, briefly centrifuge the sample to collect condensation and remove debris. For the fifth step, which is gel loading preparation, carefully remove the comb from the gel and rinse the wells with running buffer to ensure proper sample loading. Next, use a micropipette to carefully load the prepared samples into the wells, ensuring precise placement to avoid cross-contamination. Place the gel into the electrophoresis tank and fill it with running buffer. Connect the system to a power supply. And then, set the voltage to 100 to 150 voltage and run the gel for 1 to 2 hours depending on the protein size and resolution required.
Once all samples are loaded, the electrophoresis unit is connected to a power supply and voltage is applied. A typical SDS page run is performed at 100 until 150 volt. 100 volt is often used at the beginning to allow protein to migrate uniformly into the stacking gel. 120 until 150 is used once proteins enter the resolving gel for efficient separation. The electric field causes proteins to migrate from the negative electrode, which is cathode, at the top, towards the positive electrode, which is anode, at the bottom. In the stacking gel, proteins migrate freely and accumulate at the interface between the stacking and resolving gels. This ensures that all proteins start migrating into the resolving gel at the same time. In the resolving gel, proteins begin to separate based on size. Smaller proteins encounter less resistance and move faster, while larger proteins move more slowly. SDS coats protein with a uniform negative charge, ensuring that the separation is based only on molecular weight and not on charge or shape. The Lorentz buffer contains bromophenyl blue, a small dye molecule that migrates faster than the protein. This serves as a tracking dye, allowed to monitor the progress of the run. The electrophoresis is stopped on the dye front near the bottom of the gel, preventing smaller proteins from running off. After electrophoresis, the gel is removed from the tank and placed in a staining solution containing Comisi blue. This dye binds to protein but not the gel, making protein bands visible. After staining, the gel is washed in a destaining solution containing methanol and acetic acid. This step removes excess stain from the gel while retaining the bound stain on the proteins. Once the gel is fully destained, distinct protein bands appear. This will examine the migration pattern and compare the band with the protein ladder to determine the molecular weight of each protein. The thickness and intensity of the bands indicate the relative abundance of protein in the sample. Thicker bands suggest higher protein concentration, while fan bands indicate lower amount. This marks the completion of the SDS pitch process, successfully separating protein based on molecular weight. Now, we can proceed with further analysis, such as Western blotting or quantification.
Western blotting is a powerful technique used to detect specific protein in a sample. Here are the few steps in conducting Western blotting. In preparation steps, SDS page procedure being used. The protein sample is mixed with SDS buffer and heated to denature proteins, giving them a negative charge. During separation, a molecular size weight marker loaded into the gel. It consists of protein with known size that allows estimation of sizes for protein in actual samples. Each sample added to its own well in the gel. Then, electric field being applied across the gel. After proteins being separated, it being disassembled and the top portion containing the wells is cut off from the gel. When performed wet transfer, the gel equilibrated in transfer buffer. To make blotting sandwich, gel holder cassette is immersed in transfer buffer, then fiber foam pad, then same steps for filter paper. Then once the gel has been equilibrated in the transfer buffer, it being carefully placed on blotting paper. Next, layer PVDF, blotting paper, and finally fiber pad being layered, but before that, each of them must be immersed in the transfer buffer. Then, gel holder cassette closed and locked with the clamp. And the membrane is incubated with a blocking solution to prevent non-specific binding of antibodies to the membrane. The membrane is incubated with a primary antibody specific to the protein of interest. This antibody will bind to the target protein on the membrane. The membrane is washed several times with a washing buffer to remove any unbound primary antibody. Then, the membrane is incubated with a secondary antibody that is specific to the primary antibody. The membrane is washed again to remove any unbound secondary antibody. After incubation, membrane is placed in a clear plastic wrap to prevent drying and ready to be analyzed.
Now, let's look into assembling the apparatus. Place the glass plates into the casting frame. Ensure that the plates are tightly clamped together to prevent buffer leakage. Position the gel sandwich into the electrophoresis module and close the clamp system. Carefully slide the gel holder into the buffer chamber, also known as the electrode module. Insert the electrode lid correctly to ensure proper function. Verify that the electrodes align properly with the buffer chamber. It should not come off when attached properly. Connect the red positive and black negative connectors to the corresponding terminals on the power supply and set the appropriate voltage for the experiment and begin running the gel. SDS page is an essential technique for separating proteins by size using an electric field. By denaturing proteins and giving them a uniform negative charge, it ensures separation based on molecular weight. This method is widely used in protein analysis, purity assessment, and molecular biology research due to its accuracy and reliability. In conclusion, SDS page provides a simple, efficient, and accurate method for protein analysis which contributes significantly to scientific research and biotechnology application.